Hello, everybody. Good evening. How are you? Hopefully, all of you are well. Being good is a challenge at the present time. Even then, we have to be good. Okay? Because being good is our daily goal. Anyways, this is Mohammad Farooq Hussein. I'm working as an assistant professor in the Department of Physics at Joshua University of Science and Technology, Bangladesh. Today, I am here with another fantastic virtual event. So, in this stage, I am welcoming to all of you in this session. These days, we are passing a critical situation all over the world due to COVID 19. So, it's hard to organize a physical program. That's why Jamal Nozrul Islam Astronomy Club just has taken initiatives to organize various online educational programs. In its continuity, we are presenting this ninth international webinar today. And in this session, our honorable guest is Agata Maria Kołodziejczyk from Poland. Well, Agata is already connected with us. Hi, Agata, can you hear me?
Yes, I can hear you loud and clear. And good evening, everyone.
Okay, so thank you very much. And uh, we're really grateful to Agatha to be the guest of our program. We express our deep gratitude to her. Okay, Agatha, I will come back to you in a while. Okay.
Yes.
Great. Agatha Maria Kołodziejczyk. There are a lot of things to talk about her, but I am mentioning here some of her significant achievements. Like Agatha achieved her PhD from Stockholm University about the influence of light on signal transformation in neurochemical circuits of nervous systems in various animal species. She discovered an internal eyelid of insects compound eye and presented this discovery in the concurrent design facility at European Space Agency to revolutionize constructions of future modular space telescopes. She developed a prototype of lighting system to synchronize biological clocks and non-invasive treatment of jet lags. Recently, she developed a new method for in situ clothes production in space. Well, she is the director of scientific projects at Astrotech and Bioastronautics. She is an expert in the area of neurobiology, astrobiology, outer space, innovation, transformation, and transhumanism. She is the co-founder of the Analog Astronaut Training Center. She was a co-founder also of Space Garden Company and chairperson of Lunaris Scientific Council. Agatha is a board member of World Research Center and director of Advanced Concepts at private space agency, the Vallis Mariners. Okay, she is the winner of multiple prizes, such as a double winner of the best scientific experiment in the Global Space Balloon Challenge for detecting stress in biological organisms in the stratosphere. Scientific Roczak Award, a golden medal for the initiative of creating the first habitat in Poland, and even a public distinction such as one of the most powerful women in Poland. Wow. Okay, so actually, she develops acceleration of human spaceflight research in the world. She considers her most important goal not only to explore the cosmos and combine biology with astronomy, but also to inspire people. Oh, really impressive. Okay, so great. I am now sharing the poster of our program, And here you see the title of today's webinar. Okay. Oh, sorry, just a minute. Okay, so and okay, so this is the poster of this webinar, and the topic of this webinar is molecular, metabolic, and behavioral responses to starlight in organisms living on planet Earth. Okay, so I want to inform you that we have a questions and answers sections, that is Q&A section at the end of this webinar. So viewers, you can ask questions by commenting, and of course our guest will give you the right and best explanation of the questions. Okay, there was a lot of talk, so now it's time for Agata Maria Kołodziejczyk. So Agata, please carry on with your discussion. Thank you.
Can I start already?
Yeah, sure. Please carry on.
Okay, because I lost a connection. One more time, I would like to thank you, Farouk, for inviting me to this wonderful seminar, and I hope I will also inspire you astronomers, you serious guys from physics, from strict subjects, very, very important subjects. I'm the only biologist, but also I was always fascinated by mathematics, by physics, by chemistry, and I wanted to combine all these disciplines in a very complex biology, which is sometimes not considered seriously, but believe me, it is a great fun also to study biology. I will try then to share my screen, and we will go with the presentation. Yes. Can you hear now my slides? My slide?
Yeah, it is visible to us. Okay.
Super. Yes, so today we will speak about the influence of sun on living creatures on the earth. Of course, there are many influences, and the whole process, the sensitivity to light, can be discussed on many types of levels of consideration. So very basic molecular level, how molecules perceive light, how they transform. Then we have some upper layer, which is a metabolic layer, that there are some processes driven or controlled by light. And then we have a meta level of behavioral responses to light. And we will speak today about all these levels of consideration, how light influences life on planet Earth. So welcome to this lecture. Here is the main structure of this presentation. First, I will give you a brief introduction. Of course, you know the best, the solar spectrum, but I give you just some traces which are more interested for biologists and for understanding how animals are perceiving this solar spectrum. Then we will go to the molecular level, so to photosensitive molecules and systems that are observed in many organisms on Earth, starting from very primitive single cells like bacteria, algae, or cyanobacteria, photosynthetic cells. And then we will also see how these systems work in higher organisms like plants and, of course, animals, including humans. Why I wanted to tell you about this is because we are testing, my studies are about searching the influence of sunlight on circadian rhythms, which we will be talking about. And we designed a sunlight simulator to run research on humans, on plants, on animals with a particular spectra of light. And this I will also try to show you to present our results, how we designed this sunlight simulator system, how we implemented this simulator system, and then how we tested what kind of experiments we ran on plants, animals, and humans. And finally, we will get to results and to analysis. So let's start from the beginning, from the introduction about the solar spectrum. Of course, I will not go deep inside this topic because you are experts, you know exactly how starlight has, what kind of spectrum it has, what kind of characteristics this kind of light has. But here I wanted to show you some graph, some diagram showing that solar spectrum may vary depending on the weather conditions. So basically on the atmosphere that covers our planet. This is very thin but very dense in molecules layer that can modify the solar spectrum. And we see that most of wavelengths which are modulated by the atmosphere are upper wavelengths, starting from 450 nanometers until the deep red. But we see that especially this higher energetical light, shorter than 450 nanometers, is not so much disturbed. And this is very important. Why? It is important because most of organisms living on our planet, they are dependent on these wavelengths. We know a lot about how harmful UV light is because this is the range of light, blue light, violet, and ultraviolet light. But this spectrum, this range of spectrum is extremely important to run the basic processes of life on this planet. That is why I wanted to show you this graph. And here also I wanted to maybe inspire you to make some photos, like a time lapses of sunrise and sunset during a couple of days. Here is kind of a foggy day full of clouds, but still we can see changes during the sunset and sunrise. On the left side, from the dark to the bright color, you see the photographs that they were taken at dawn at intervals of 15 minutes. And you see that the color of sky is changing, and it is changing quite rapidly at this specific time. We see also a high level of blue light during the sunrise, and this is what I really wanted also to indicate you. Maybe some of you are not so much familiar with this phenomenon. So blue light is very important in the morning, and it is inducing several reactions inside organisms of living creatures. So this blue light is important. We see also on the right side the photographs taken at dusk, again at intervals of 15 minutes. And you can also see changes in the intensity of color of this lighting. This work was done by my friend. She is a photographer, professional photographer, and we wanted to visualize these rapid changes in the color of lighting of sunlight. For sure, that are related to light, that are really sensitive to light. And for example, you see this field of sunflowers, and you see that all flowers are turned into the same direction, toward the sun. If you will look more carefully, these flowers follow the sun during the whole day. Even if there is a cloudy day, they sort of follow the sun. So they have some receptors to follow the sun, and they have some more complex systems because rather a complex system dependent on light. And this is fantastic. Already Linnaeus, who named several species on planet Earth, he also observed that sunflowers, they are opening, these flowers, they are opened at proper time of the day. And he even could design a flower clock showing which species open their flowers during the daytime. So again, we see that such, we can say, simple organisms like plants, they can sense the light in a proper manner. So they can even transform this light information into time information. And of course, human visual system of many more advanced organisms is a specific organ to detect light, various wavelengths. So it is not only detecting the light, but also detecting a specific wavelengths. There are specific receptors, which I will say in a moment, that they can feel the light and they transform this information into very complex processes inside the body. So first, we will start from photosensitive receptors. Of course, photosensitivity is a very, very old phenomenon in nature, and it began just when life began on our planet. So it is very ancient phenomenon. One of interesting receptors that are photoreceptors, and they are most common receptors, these are quite complex G protein coupled receptors. They have seven membranes, seven transmembrane domains, that are mounted in the membrane, cellular membrane of the cell. And you can see on the left our human eye with the retina, and you can see several layers of photoreceptor cells. And these photoreceptor cells, they contain multiple types of GPCRs, we call them. And many GPCRs react to light. They are called rhodopsins, or rhodopsin, which is a protein called opsin, and it's found in the retina of many types of eyes. GPCRs, they also respond to olfactory stimuli. So they are also different types, but mostly I wanted to focus on rhodopsins. And in the brain, this family of receptors is involved in transmitting dopamine and serotonin signals. In the nervous system, they control automatic bodily functions such as blood pressure, digestion. It is also interesting to note that insects and birds can see UV light, so there are special GPCR receptors to detect UV light. And humans, we also have UV rhodopsin, but we cannot see is this signal directly goes to the brain without conscious perception. So this is also important, that our eye can see much more that we consciously can detect. Very interesting thing. Why we perceive UV light? I hope I will answer you during this lecture. One of vitamins, vitamin A, which is known as retinol and retinyl ester, they are sensitive at 325 nanometers. Vitamin E is sensitive at 292 nanometers. Carotenoids, which are also present in human body, they are sensitive at 450 nanometers. So look, again, these all wavelengths are these higher energy types of wavelengths of solar spectrum. Another very interesting group of photoreceptors are cryptochromes. I strongly encourage you to search more on internet or in some handbooks about cryptochromes. They are amazing molecules which are UV and blue light photoreceptors. They are similarly structurally to very old photoreceptors, photolyases. And this is a class of DNA repair enzymes. Most of you probably heard that UV light is harmful, it can break your DNA. But also there are special mechanisms that protect DNA from UV light. And these are DNA repair enzymes, cryptochromes. For example, in birds, cryptochromes are essential compounds in migratory flights. They are related to the perception of the Earth's magnetic field. So without UV radiation, birds cannot analyze the planet's magnetism. Can you imagine that all this fantastic sense of feeling the planet's magnetism is dependent on cryptochrome photoreceptor to UV light? The maximum absorption for cryptochromes is 380 nanometers. And again, I want to highlight that cryptochromes are very old molecules. They began very early in evolution of life. So they occur in most of biological clocks of almost all living creatures in the world. So this is fantastic that we all have the same type of molecules in our bodies. Much more advanced molecule and still very, I would say, very enigmatic, very mysterious molecule is so-called melanin. You know melanin because this is a pigment of skin, very popular in everywhere in common life. But these melanins are quite complex polymers whose chemical composition depends primarily on the nature of the substrate and the local conditions in which these substances are formed. So it is not easy to strictly say what is the structure of melanin. And in humans, melanins are found in the skin, as I told you, in melanocytes, and they are characterized by white range of light absorption. They are characterized by a wide range of light absorption, again, especially in short wave ranges. Melanin is found not only in humans, but also in bacteria, fungi, plants, and other animals. So even it is so complex molecule, it is again observed across all types of living creatures on our planet. In cases of too intense exposure of UV radiation, we can expect that some melanocytes will transform into a cancer, a melanoma cancer, which can be very, very dangerous. So every year we should look at our body if we do not have anything growing fast, which is pigmented, usually dark, but it is not. It shouldn't. It is sometimes that there is no strong pigmentation of this cancer. And here I wanted also to mention you that this melanoma cancer can occur more often if babies are exposed to the sun, to intense radiation of the sun. Later on, these kind of effects are less harmful. So also, if we are exposed to the sunlight in the morning, then our body is prepared for UV light and we have all protective mechanisms activated. But exposure after noon, it can be much more harmful because our protective systems are shut down. So it is very important to have this knowledge when we should go outside and be exposed to the sun to be healthy and to protect our bodies. Finally, another type of photoreceptors, antioxidants, can be characterized. They are also sensitive to light. These are natural substances observed in several fruits, vegetables, plants, of course. So all types of vitamins, E, C, beta-carotene, some anthocyanins, flavonoids. They have this feature that they can capture free radicals, and yes, they are very wide present everywhere. Of course, I have to mention vitamin D, which is responsible for a proper bone growth and many other physiological processes in the body. The synthesis of a vitamin D3 in the skin depends on factors such as latitude, sun exposure, time of year and day. So the angle of the sun that touches our body. Of course, the cloud cover thickness, air pollution level, exposed skin area, skin type, and body weight. So there are so many parameters dependent on this vitamin D synthesis in the body that it is hard to say to all people, you need to go outside 20 minutes per day and it will be enough to get the proper vitamin D level in your body. It is impossible to say like this. But what we know is that, for example, in latitudes above the 37th parallel from November to February, there is a significant reduction in the amount of UVB light, UVB photons reaching the Earth, and then of course we need to somehow to take vitamins using drugs because we need to take them from diet and not from light directly. Of course, skin pigmentation plays an important role. More darker skin you have, so more melanin you have in the body, you need to be exposed more to the sun in order to get the proper vitamin D levels. So sometimes people that have darker skin, they have depression and they don't understand why. It is because they need much more sun than people like me with relatively low melanin level in the skin. Another feature is that older people, they cannot synthesize vitamin D so efficiently. So again, here they need to add some vitamin D into diet. For sure, you know chlorophylls, so I will also mention them here. Of course, plants, they feed on solar energy, on light, using two types of chlorophylls, which are molecules that can transform light photons into energy. And this is chlorophyll B. You can see the profile of absorption, and chlorophyll A. These types of photopigments are in chloroplasts, special structures inside the plant cells. And finally, my best topic, which are biological clocks. I was always, I don't remember when, I was amazed and fascinated by biological clock and how this clock can be tuned or set by light. So, what is biological clock? Most of living organisms have adapted to the circadian, so about 24 hours day and night cycles. Circadian means about 24 hours. So most of animals, of course, they had to adjust to this rotation of our planet. And this effect of the adaptation led to creation of molecular biological clocks. And these are cyclical changes in genes that they have effects on physiological processes over time, and they determine the phases of activity and sleep. So our activity phase is not just that it is sunlight and we are activated directly by light, but this is far more complex. This same for sleep. It is not that there is no light, we are bored, we will go to the bed. No, it is again far more complex process. And all these observed changes, how we behave, are indirect result of the expression of biological clock genes. And these biological clock genes cyclically release proteins, so they are activated, these genes, and they express proteins. And the increased amount of these proteins inside the cell triggers the synthesis of negatively related proteins. That is, proteins that inhibit their expression. So there are negative feedback loops, which are the basic mechanisms of this molecular clock. And here I wanted to show you the table of different types of biological clock rhythms. Of course, they are caused by movement of celestial bodies. First, we have Earth's rotation, And we have circadian cycles, which are 12.4 hours long. And for example, the circadian rhythms, they are visualized by mollusks feeding behavior. So we can observe that they feed in this kind of cycles. Another type of the cycle is 24 cycle, which we already mentioned. It is circadian cycle, and it is caused also by Earth's rotation. And this is very, very usual. We all have it. It is a sleep-wake cycle. We have also very interesting cycle, which is caused by moon, which is a moon's revolution-driven cycle. And this is a circalunar cycle. It has about 29 days, and it is observed in female, in humans, in females, in women, and it is just a menstrual cycle, which is a circalunar cycle. Very interesting. Finally, we have also a rhythm driven by Earth's revolution, which is 365 days, circannual. And many animals, their reproductive systems, they have these circannual rhythms. For example, rutting in deer. So this is very interesting also to see how many clocks we have inside our body. These clocks were observed quite early by Desmarais. Here is the experiment that he performed. So you see a plant that was covered from the sun, and in constant darkness conditions, this researcher observed the same rhythms of leaf movement. So during the day phase, even it was dark, leaves were opened, exposed to the light, and during the night phase, they were closed. So all rhythms that can be observed in constant darkness conditions, these definitely are driven by circadian rhythm, by biological clock. And here is also some graph, some diagram visualizing that we do not have only one circadian rhythm, which is awake and sleep phase, activity and sleep phase, but we have much more time points during the day and night where our genes are activated. What I told you with vitamin D synthesis. When you will be exposed to the sun in the morning, you will synthesize the most efficiently vitamin D because this is the time for it. The same, we have time for many, many things, like for example, for taking drugs, for digestion, for physical training, and many, many, many others. All these metabolic changes, they are driven directly by biological clock. So we can say that we are rhythmic creatures. Where is our biological clock located, you may ask? It is this upper graph. What you can see here, it's a schematic of our brain connected with eye. So we see light passing through the eye, through the retina, and it goes to different centers, different types of clocks that are located and connected in the middle of the brain. The main clock is located in suprachiasmatic nucleus. So this is this blue one, the most dominant clock, and then this clock is connected with other types of clocks. Why we have also other types of clocks? It is because of security reason. We cannot have only one system, and then if one system will be broken by many reasons, then we will lose such a basic and important mechanism like biological clock. So biological clocks are located autonomously not only in the brain but also in the skin, in some our organs, in guts, etc. And this is showing how important is biological clock. And most of clock studies, especially those that they got a Nobel Prize in 2018, because there was a Nobel Prize for biological clock, for discovery of this clock in organism, most of research was made on a model animal, which was a fruit fly, Drosophila melanogaster. And also I was making my PhD on Drosophila visual system. Visual system in Drosophila takes 2/3 of the brain. So it is the dominant part of the brain. And you see also here below, in the bottom, you see how many clocks Drosophila has. So not only in the brain, you can see this Drosophila body drawing. There is one clock in the brain. There are separated clocks in Drosophila's eyes, in olfactory bulbs, then in the body, in abdomen, even in legs, in the cuticula, in skin. So again, this shows how important biological clock is. And all these clocks are related, are dependent on light. This is again very important. Look how many things and cells are important, are dependent from sunlight. So how is it going? I told you about negative feedback loops that are the basic mechanism of the biological clock. So we can start from more clock gene mRNA is made. So first we have some light that induces, activates the gene, and then gene is activating protein production. So more clock proteins are made. Then clock proteins enter nucleus, so they are moving inside the cell to the nucleus, and clock protein inhibits expression of clock genes. And then we have a less clock gene mRNA, and clock protein levels is decreased. And it goes again and again and again, the same way around. Of course, it is not so easy. We have several clock genes, not only the one. We have many related, directly or indirectly, proteins that all build this complex and beautiful mechanism of clock inside our bodies. So maybe here I will skip because we maybe don't have time so much, but here I wanted to show that here you see this stripe, night, day, and continuous darkness. You see this on this graph. And we see that the main characteristic of circadian rhythm is the amplitude, the phase, and the period. So this is quite similar, like in many processes described in physics, even in astronomy. And we see that this circadian rhythm is not changing when the light is off. This is very important. So what happens if there will be one guy living many years in the cave, isolated from natural light? Well, it will still keep circadian rhythms. but it may feel that days are going much shorter. So after, say, 20 years, we know that it was 20 years that he was in the cave, but he would think that he was, say, maybe 15 years only. Circadian rhythms may be reset. This is very important, especially if you want to modulate the circadian rhythm. So you can reset the circadian rhythm by using, of course, light. And depending when you start to give this light signal at which phase of the rhythm, then you can expect different changes in later spectrum of your cycles. So this is also very interesting how to do this. And now I will go to our research after this basic introduction. What happened now when we have electrified planet where most of people have lamps, artificial lamps inside their houses, their offices, their shops, hospitals. So we started to use not only sunlight, but we started to use electricity in our lives. What this caused? What kind of change occurred after electrification and urbanization? Well, I must tell you that this is very slow process, but we see that there is a tendency that people are living more and more inside their homes. They are more and more isolated from sunlight, and they are mostly using artificial light. The question is if this artificial light is sufficient to keep all our molecular and metabolic processes in a healthy state. Yes, because this is very important. In order to do this, we designed a sunlight simulator light, but we were mostly focused on these wavelengths that do not exist in standard artificial lightings. So all these LED lamps that you have at the university, everywhere, all bulbs, they do not have the UV light spectrum. They do not have a blue light spectrum at these wavelengths that I mentioned you here. So we designed the light, now it is very easy to do this because we have a proper LED technology. We can select which wavelength of LEDs we want to implement in such a system. So we implemented these kind of wavelengths, and we tried to see what will happen, what are changes between natural light exposure, our sunlight simulator exposure, and artificial light, regular standard light exposure. So again, I wanted to highlight here that important aspect in the design of this sunlight simulator was analysis of LED types available on the global market. And sometimes it was not so easy to select the proper UV LEDs because their characteristics was not exactly what we wanted to achieve. But fortunately, every year brings better and cheaper solutions to obtain optimal assembly. So for all of you who are interested in experimenting, who have some engineering skills, who like to do this, I strongly recommend you to design something like sunlight simulator system and try if this light is maybe not better than standard artificial light which we are using. Important aspect in the design phase is searching for those limits, considering safety issues. So, of course, this is very important. It is important to keep light functionality and to keep this light safe. Due to the special importance of UV radiation and its key role in the project, here I only remind you that we did not use UVC radiation. This UVC radiation naturally does not exist on the surface of our planet. It is fully absorbed by ozone layer in the stratosphere. What it gets to our surface of the Earth is UVB radiation and UVA radiation. Also, very important to note is that UVB radiation, these wavelengths, this range of spectrum, usually is not transparent for windows. So windows, they are absorbing UVB radiation, and even we are sitting next to the window, our body is not getting UVB radiation. This is important because still window is not protecting us against low levels of serotonin in the brain and depression. And we are also not synthesizing vitamin D by sitting next to the window. Okay, so this was our design, the main table representing different types of modes that we designed. So we designed a special light cocktail. We named it for plant cultivation. Here you have in the left column. So you see what kind of types of light we used, what kind of wavelengths, and what kind of proportion in the whole cocktail of the light. The auto mode we designed, of course, these all cocktails that I present here you on the table, they were made based on available literature. So nothing is completely new. If you are only interested in designing sunlight simulator, you have all knowledge, all information available in publications. And for work, of course, we added more blue light. We added, less warm light. For relax, we got more red, but less blue light, and so on and so on. For seasons, we are not showing here the percentage of different wavelengths because they are changing depending on the time. And energy boost is a typical boost of UVB, UV, royal blue, and cold light, which is more than 6,000 kelvins. This is also very important, the temperature of light. Okay, then we changed the design from these blue light type lamps, because these lamps, they were mostly blue light emitting lamps. And we tried to design lamps that can be used normally everywhere. So in the hospital, at universities, at malls, big shops, factories, etc., where people are working usually many hours in the artificial light. So here you see implementation of the light that was designed. We designed this together with a QLab company. This QLab company was specialized in lighting. So I only told them what I want. And here on the right down, you see the metal box with some display. This is a touchpad, and you see 5 channels, 5 modulators of light. I'm sorry you probably can't see, but I can tell you from the left, you have UVB light, UVA light, you have visual light, you have temperature of light, and then on the right column, you have infrared light. Why infrared? Well, this infrared panel we wanted to have because there is not a lot of literature available about the influence of infrared light on organisms. So when we designed this lamp, we wanted not only to try and test influence of UV light on some processes, but also we wanted to detect maybe some effects with exposure of infrared light. This place, this building is our habitat. So this is isolated home. We isolated it completely from sunlight, and we close people here for many days so we can observe circadian rhythms. As I told you and showed you before, circadian rhythms are also present in darkness. For us, this artificial surrounding, artificial environment is treated like a darkness. And for biological clock, it is very good. So this is kind of a biological clock laboratory. But first, we wanted to test if our light, especially UVB range and UVA, but mostly UVB, if this range of light is not harmful. So before we started experiments on humans and on animals, we wanted to see if this light is maybe not harmful. And we tested various types of plants. Here I only show you salad and cress. And we see on B, in case of salad, you see that some of these seeds did not develop properly. So leaves are closed and they are damaged in a way that they couldn't develop later on. So these plants stopped to grow. They were sort of burned by UVB light. In C, you have control, so you see how it should look like. In A, we have exposition to natural sunlight, but because it was very cold outside, where these plants were growing, we do not see the effect. This control C was growing next to the window. So we see that plants do not need UVB light to grow properly. In case of animals, we made tests on fruit flies. I showed you the brain of fruit fly and the biological clocks of fruit fly. These fruit flies are located here in these tubes. These are polypropylene tubes, and they were exposed to light in the paper box, so isolated from external environment. Of course, we measured temperature, all environmental parameters were constant, and only we used again UVB light. Why? Because we wanted to see what will be the mortality of these fruit flies comparing to flies living in natural light and in artificial light. And finally, sorry for this slide, we made experiments on humans. So once we set safe parameters for our UVB, UVA channels of light, we recruited people for our experiments. And the human studies were adapted to the analog astronaut training center habitat, which I showed you before with these lamps. Additionally, we put sensors to measure all environmental parameters. So we really had ability to measure humidity inside the habitat, temperature, pressure, CO2 concentration, light intensity, electricity consumption, etc. And well, we had several groups of people. To make long story short, these were young people, 18 to 39 years, and the first teams, Bright 1 and Bright 2, they performed the longest experiments. So first 2 weeks exposition was only for white artificial light without UVA, UVB, and infrared. So this was a control mission, because this time inside the habitat we named mission, and people they had some tasks to do, and they all were on the same diet, etc. And then the same crews, they spent 2 weeks exposition of white light plus infrared, so no UVB, no UVA. And this infrared was only administered 100% for 3 hours per day. So when they were eating at the table, all the crew was at the table, and then they had this infrared exposure. And same was for the last type of experiment, 2 weeks exposition of white light plus UVA plus UVB. And then again, it was only 3 hours per day during meal times. At 9 a.m., at 1 p.m., and at 6 p.m. Then we had another crew where we tested how this UVA, UVB light is important for treatment of jet lags. You probably know what jet lag is when you travel abroad, especially with trans-Pacific flights, transcontinental flights. You cross several time zones, and of course you have jet lags. So we wondered if there are people working on shifts, like many people are working on shifts, especially in hospitals, in factories. They also suffer from desynchronization of biological clock. And this is very dangerous for health and for aging, because these people, they age much faster. So we had this hypothesis that our lighting may treat these jet lags. And then we had another crew, which was a crew when we had only men, because we wanted to measure the testosterone levels and exposition to UVA and UVB light. And finally, we had 2 crews with people with different melanin levels in the skin. So we also were wondering if this light is influencing white skin people, or maybe also some darker skin people too. And we wanted to see how big changes we have. Again, this is our Analog Astronaut Training Center habitat, and these are our crews. You see on the upper row left, starting from the left, Bright 1 crew, then Bright 2 crew, then Eternity crew, Destiny crew, Ample 1, Ample 2. In case of Bright missions, we had to collect blood samples. So this was a big sacrifice of our analog astronauts. And here you see down an analog astronaut from Bright 1 crew who is collecting blood from somebody, some of their friends. Yes, and also on the right down, I'm showing you our screen of parameters of the habitat sensors, and how it looks like during the mission. And here are some examples of our results. We made ELISA tests, which are immunocytochemical tests sensing serotonin, cortisol, melatonin, and testosterone. The method used was that we distributed samples and coded the samples that you can see on the upper table, and then these codes we measured 3 times. We measured luminescence and absorption of the sample, and we computed the average from 3 measurements. So here I only want to show you the data from serotonin, cortisol, melatonin, and this red means that this is from blood, white from saliva, and yellow from urine. So we tried to visualize results of the same substance in different kinds of solutions. Sorry for Polish language here in tables, but here I only wanted to show you our results for cortisol, which is the upper line. On the left, we have a cortisol in the blood, and we have a control mission, experimental mission with infrared, and experimental mission with UV. The same cortisol substance in saliva visualizes completely different numbers, what you can see in this upper line in the middle. These are averages, and the error is computed. So we have again a control mission with infrared, and mission with UV. So it looks like opposite. And then on the right upper line, we have cortisol in urine. Again, different things. So I want to show you these results, not only to show how many results we did, but how difficult it is to to analyze this data. There are completely different values and concentrations in different solutions. Of course, we know indirectly how to analyze this data, but it is not very easy. And same we have for melatonin. Melatonin is a characteristic, we name it like a neurohormone of sleep. For example, astronauts, they have pills, melatonin pills, to be able to sleep on International Space Station. And many people who suffer from insomnia, who cannot sleep, they are also, they have these drugs, melatonin drugs. But melatonin is a natural neurohormone synthesized in the brain, and we have the highest levels of melatonin afternoon, in the evening before sleep. That is why we feel sleepy, and that is why we want to go to bed. But many people have disturbed melatonin, especially, probably you heard about this discovery, this research, that when you are in the bed and you are taking your smartphone without blue filter, then you block through your photoreceptors, you block synthesis of melatonin in the brain. So you are not feeling sleepy. You can still watch the movie. You can still work in front of your laptop. That is why very important is to download blue light filters on your electronic devices, especially before going to bed, 2 hours before going to bed. You should have this red screen in front of your eyes. And the lowest line here is for serotonin. I am a very big fan of this neuroamine. Serotonin is synthesized in the brain. It is synthesized, of course, in the visual system. And serotonin is commonly named as a neurohormone of happiness, a molecule of happiness. But in fact, this molecule is responsible for your activity state, for how open to the external environment you are. So this means that if you have low level of serotonin, you suffer from so-called depression, and you only see your body, yourself. You are not looking at other people, at flowers, at trees. This is not important for you because you have low level of serotonin. But if you have a large level of serotonin, you are opposite. You are in a mania state. So you want to do everything with others. You are so happy, jumping to the sky. And again, you see that we have different values, different concentrations in the blood on the left, the lowest line, in saliva, the middle diagram, and in urine. For testosterone, again, we saw that control mission, then in the middle you have experimental mission with infrared light, and then on the right, experimental mission with UV light. We see that this testosterone is also sensitive to the type of lighting. We analyzed vitamin D synthesis in response to our sunlight simulator lamp. We used commonly available on the market vitamin D test. All people around the world can order this test, I guess, but this is not advertisement. We did simply this test. It is the most simple to be done, and we observed again changes. So comparing to regular standard artificial light, our light with UV ranges, it caused advanced synthesis of vitamin D in the body. This is very good. We also looked at circadian rhythms. Here I only show you how we did it. So we had an application on laptops, and each analog astronaut, was supposed to register just after waking up and just before going to sleep, not to bed, but to sleep. And then we asked about the mood and about temperature and heart rate. Based on this information, we created very well known in chronobiology, so the biology of clock, actinograms that show how long you were active and how long you were sleeping during the day, so 24 hours. And each line corresponds to the day of the mission, from 1 to 12, because these were like usually 2 weeks missions. And we observed that in the habitat with our lighting system, these biological rhythms were very regular. This means that the metabolism, that all behavior was very regular and harmonic. We also analyzed water metabolism, and here you have an example. So, for example, we have 4 people: commander of the mission, astrobiologist, crew medical officer, communication officer. These were participants of the mission. You have mission days, and each person was supposed to write and report how much water she drank, how much urine she peed, and shower. This was for technical water report, so it is not important here. Again, sorry for Polish description, but here we see the water balance, where we have the drank water, which is depicted in the blue color, and urine in yellow. So we see that in control mission, look please at the upper diagram. On the left, we see comparison drinking water to urine in control mission, and then we have the same proportions for experimental mission with infrared and experimental mission with UV light. So comparing to artificial light, it is different. It is, it is different. Based on these results, we can say that, again, water balance, so metabolism, is regulated by light. Same for sleep monitoring. We observed that sleep in these conditions was really good. So melatonin levels were high enough in order to get good quality of sleep. We measured physical activity. Here you see how it looked like. So we had the average body parameters. We measured body temperature before, after training, heart rate before, after training. Why? Because we wanted to see the motivation factor. More people are tired. It means that they train more. They are more motivated to train. And of course, we have standard parameters like time of workout, distance, and burn calories. So I'm coming briefly to results, because there were many data that we correlated with each other, and it is of course too short time to explain everything, and anyway it would be very boring. So I want to present you briefly analysis of behavioral, morphological, biochemical, and molecular changes that occurred in the tested organisms under the influence of various types of lighting. And we also tested these harmfulness effects associated with long-term use of sunlight simulator. Again, sorry for this slide, but here are some of results which are important for us, for humans. So here we observed that there were no behavioral changes. At least they were not observed negative behavioral changes exhibited in a negative impact of lighting. So we observed only increased concentration while working. People who were isolated from sunlight did not feel the need to go out to outside the habitat to be exposed to the real sunlight. So we did not observe this. People reported that the habitat's solar simulator did not resemble sunlight due to the lack of variation in intensity and color during the day. So they told us that they really wanted to have some variations in lighting, and the light was constant, so it was like artificial light. But they felt very concentrated, and they did not feel any negative emotions, any negative effects. Also, we did not observe any morphological changes, for example, change of skin color. You know that after UVB exposure, we should be sunburned, but we did not observe in our analog astronauts anything like this. So this means that this UV was limited, and it was too low level of UV light to get this sunburn. And finally, the analysis of biochemical and molecular changes showed a positive effect on the synthesis of key compounds of the biological clock and the hormones, cortisol, melatonin, serotonin, and testosterone. Why I say positive effect? For me, positive effect means that there is effect. At this moment, we are not able to determine if this is really healthy or not. So positive in a definition of healthy or not, this I can't say now, but I see that there are changes, so the effect is positive. I hope it is clear. And we observed a significant decrease in the synthesis in compared conditions. Okay. We also observed positive effect of lighting on the water balance and vitamin D synthesis. So this effect, the positive effect, so we see that there was something induced by light. It occurred even after 20-minute exposure of our sunlight simulator. What means that this is enough for, of course, white skin people that are more sensitive to UV light. What else I can add here? Well, about jet lags, I can say that we observed positive effect of sunlight simulator lighting system. So adding UV ranges of light induced faster recovery after jet lag. So it was quick recovery, 1 to 2 days after the phase shift. People felt completely well. And for plants and fruit flies, we just made some investigation about limits of using our light and to develop some safety limits. So we observed that, of course, we shouldn't use UV light 100% 12 hours a day. And we shouldn't use this lamp in closer distance than 45 centimeters. So also the light intensity is crucial in these damage features. The final conclusion is that the lighting conditions of living organisms have a huge impact on the quality of life. And I think that all we agree with this. So this is like breathing. This is like a good bed, like good shoes. We need to live in light in order to work efficiently, in order to be who we are. So I think that we all should take care of our air, of what we eat, and of our light. Especially important is to know that long-term exposure to artificial lighting can induce a negative impact on health due to the shortage of numerous photosensitive substances because they are synthesized only in the presence of UV radiation. And this UV radiation is usually impervious to glass, to windows. So this is very important. And the tested lighting prototype meets the basic assumptions of natural light simulation. So we extended the emission band, not only visible light, but also we expanded this emission band to the violet and to infrared. But we observed that infrared did not have any effect. Not maybe it had, but not so much as UV light. Still, we need to do many studies and to really understand what is going on inside. And we believe that this type of lighting, which you can all create by your own, just buy a proper LEDs and cover the spectrum of the sun as much as you can. Then we believe that this kind of thinking about light should be very helpful in curing depression, in curing all these low levels of photosensitive proteins in our body. We are powered by light, and if we kill our photoreceptors, if they will be not usable, then I think that, yeah, our efficiency will lower down too. And I think that with this, I would like to acknowledge, of course, all people who helped me to present these results. So engineers, constructors of the prototypes of lighting, QLab company, all people who sponsored this project. I would like to thank all analog astronauts that helped to test this lighting system and all researchers that participated in data analysis, because it was a hell of work. Here I just show references, so if somebody will be interested later on, I can share it with you. And with that, I would like to thank you all for listening, for being with me. I hope you enjoyed the lecture. Maybe something was new for you. And I invite you to collaborate with us if you have any interesting ideas or tests which you would like to do in our habitat or with our very clever analog astronauts. You are more than welcome. Thank you very much.
Okay, that's good. And thank you, Agata Maria, for your clear and amazing presentation. Okay, so
Thank you.
right now, yeah, okay, you are welcome. So I think right now we can start our questions and answers section. Okay, so we are getting a lot of questions basically from the viewers. Okay, so I think you are ready. Okay, so the first question from that was the first questions, and this is from Al Mujahid Afridi. Okay, so this is the question here. So, don't you think some analyzed microfungi and their cells are difficult to break for a DNA extraction? Okay, so this is the question. Okay,
Yes.
so, yeah, please, yeah.
Well, depending on what method you are using for breaking. But I saw one type of melanized microfungi, and they are growing even in response to ionized radiation. So they are very promising microorganisms to create a protective shields for future spaceships and bases on the moon and on Mars. So, well, I don't know if they are difficult to break DNA extraction, but definitely they are very, very extreme organisms and very hard to kill in this way.
Okay, thank you. So there is more questions here. There are more questions here. Okay. So another one, and this is the second question, and this is from Nasrin, of course. And the question is, how does the sunlight help in producing vitamin D in our body?
So vitamin D is produced in the lower layers of our skin. So not directly in the upper layers, but a little bit deeper. And this is simply the conversion of the molecule of vitamin D. And this conversion is, you said, sorry, in producing vitamin D. How does the sunlight, sorry. Yes, it activates an unactivated form of vitamin D. And this is again the feedback loop. So, of course, this is very complex process. I don't know by heart, but definitely you can read about how light is inducing the synthesis of proper genes, and how, of course, light is inducing the transformation of inactive vitamin D into active vitamin D, because this is like there are several processes involved in this process.
Okay, thank you very much for your answer. Right now, I am picking another question, and this is from Ali Murtuja, and this is from Ali, and here is the question. And okay, so there he said that we used to apply mustard oil while on our body and expose ourselves under sun in winter to get vitamin D. Is it effective? Can we get vitamin D from sun in summer, where the temperature is very high, like 37 degrees Celsius? Okay, so in that case, uh, how we will get vitamin D? Okay, thank you.
Thank you for this question. Of course, vitamin D synthesis um, is activated by light, no matter if it is winter time or summertime, because this is dependent on the wavelength, not on the temperature. So, of course, temperature may speed up some processes because all molecules are, of course, moving faster. This is just the kinetics of molecules. So, of course, this can happen faster. But the main evoking factor is light and not temperature. And you are asking if using oil is effective or not, maybe, because I don't know. You are asking about winter and you are adding a mustard oil. Well, I think that oil is not really helpful. It is only making your skin more, how to say, protected from other types of wavelengths. And it prevents drying the skin.
Okay, thank you. And right now there is another question, and this is from Al Mujahid Afridi, and he said his question is why normal human melanocytes melanin production is stopped after passage 6.
Well, I never was growing melanocytes, so congratulations. But maybe think exactly about the wavelength of light that you are using. Maybe they simply die because they have no light, no proper wavelength.
Okay. Thank you for your answer. And this is another one, and this is from Astin, of course. What is the mechanisms for melanin to protect sunlight in our body?
I think it is the similar process like in case of anthocyanins or beta carotenes, that they are scavenging free radicals in our body. So maybe this is the main protection mechanism.
Okay, thank you. And this is another one, and the questioner is Ali again. And we asked what was the process of creation of first organism on our planet?
Very beautiful question, but I cannot answer this. Well, there are many hypotheses. I remember that we were sitting at some seminar and we were thinking what was first, serotonin or serotonin receptor, like egg and hen. Ooh, well, it's very hard to say, but definitely there must be some energy in order to empower any kind of process. I think that first we believe that it was some molecule to protect some information like ribonucleic acid, and maybe the first process was just to protect this information, but it's very hard to answer because there are many hypotheses.
Yeah, so thank you. And next, another question. This is from sulfuric acid. And his question is will the new generation of astronomical instruments ever reach the farthest corners of the universe? Yeah,
Ooh.
okay.
Well, I think it is hard to see something from one point. It is better to go there and to see directly. So I think the best option would be to discover some transport to the farthest corners of the universe, and then just to have a communication sufficient enough in order to get this information from very, very far.
Okay, thank you, Agata. And this is another one, and this is from Hassan Shaharia Fahim. And is every people have the same biological clock? Okay, this is.
Thank you for this question. Yes, yes, I think we all humans, and not only humans, all animals, we are all sort of the same. The main idea of the biological clock is the same. Of course, we might have differences in genes, but again, we have the same feedback, negative feedback loops. We have some conserved, very well conserved proteins, like I told you about cryptochromes. These we have all: bacteria, plants, animals, humans. And some of these molecules we share all exactly the same, but some they are more fancy one. So biological clock itself is the same. We all are powered by clocks, and so this is the same, but some molecules, like some designs are different.
Okay, thank you for your wonderful explanation. Okay, so this is a question from Shubham Future, and he wants to know what is the comparison of the effect of sunlight on anaerobic and aerobic organism.
Oof. Very
Okay,
interesting.
this is a tough one, right?
This is my first question in my life, but very nice. Oh my God, I need to remember this. Very interesting question. Usually, anaerobic organisms, they live without sunlight, somewhere deep underground, underwater, where there is no air. So then these organisms, they do not have biological clocks, but because they don't need them. So they are not sensitive to light in this way. That is why there is a change, a big change in metabolic mechanisms which are controlled. In case of anaerobic organisms, I think that the main system controlling all metabolic states is simple: the chemical environment, the external environment, which is not light, of course, but which is for example, temperature, for example, some chemical composition, pH, something like this. That this will be this setting the factor of metabolism. In case, of course, of aerobic organisms which are exposed to sunlight, then the main factor is the light.
Okay, thank you. And then the question is from Ben Hu. And the question is it possible to measure extracellular and intracellular melanin?
Well, I guess that in case of some smaller organisms like fungi, let's say, maybe we can extract these melanins because they are different. Extracellular melanin is different than intracellular. So based on the density of the molecule, the mass of the molecule, maybe we can extract these two types of molecules and then to measure the quantity or quality. Yeah.
Okay, thank you for your explanation. Okay, so this is another one, and this is from an Astrid approach. What do you mean by directly and indirectly protein? That is direct and indirect protein. Okay.
Probably, yes, I missed something. So there are some proteins that are directly somehow activating other types of proteins. So, for example, there is, as I told you, serotonin and serotonin receptors, and they fit to each other. So they can bound together, and then they change the conformation, molecular conformation, and then they are causing some reaction. So these are like direct interaction of proteins, and indirect interaction of proteins is, for example, that there is, for example, a cryptochrome which is somewhere in the cell, and it binds to another molecule, and this dimer of molecules goes to the gene and activates another protein. So this is indirect interaction with the protein. I hope you understand what I mean. But yes, I am a biologist, and sometimes I'm not speaking very logic. Sorry, it's different education.
Okay, so thank you. And this question is from Moid Ahmed Sakib, and he said that there are some other stars that we can see from Earth. Is there any effect on the living organism on Earth of these lights emitted from those stars? Okay, so this is the question from Sakib.
Well, I think that now people are not sleeping exposed to stars. So we are not sleeping under million stars hotel. The thing is that I think that the light coming from other stars is too weak with its intensity, that it does not really activate our photoreceptors. And of course, we see the starlight, and some of photoreceptors are still perceiving this light. So this is for sure. But the power, the intensity of this light is too low to really activate the whole machinery. However, my professor, from the expert from Drosophila flies, she said that even few photons can reset the clock. So that is why we used to work in only red light, because flies do not see red light. They only see UV light and green, but they don't see red. So when we wanted to keep them in constant darkness, in fact, we were keeping them in red color because they did not see this. And their clocks were like completely blind to this red light. Yeah, I don't know, honestly. No one has been studying the effect of other stars to human brain.
Okay, so this can be another one for you to investigate in future. Okay, all right. Okay, so thank you. And this question is from Maibur Rahman, and he is really excited about this webinar, and that's why he gave you a special thanks. And he said, "Thank you, ma'am, for your extraordinary presentation. I just want to know about the artificial sun project." Okay, so and this is running by some organization. Isn't it creating a bad effect on the biological clock of us or other living species of Earth?
Well, it is like with everything. It depends how you use this technology, how you use, this product. So I believe that the most important is education and understanding what really this artificial sun project is doing and what kind of effects can be achieved in a way, and then just to be wise and use it wise.
Okay, so thank you. If you wish, we can pick up more questions because there are a lot of questions. So can we pick another one? Okay, or I think
you can send me via email, and I can answer you, and then you can distribute this somewhere. Okay?
Okay, thank you. So this is the last one, I think, and this is very interesting. This is an interesting question. Basically, this is not a question actually. It's a proposal. So I think, and this is from Hassan Shaharia Fahim, and he he is interested to work with you. So,
Thank you.
okay, yeah. So please tell something because especially uh, there are many students who are watching this webinar with us. So actually, I think he is very fascinated about your work, about your activities, and that's why I think he's interested to work with you. So I
Yeah.
think.
Of course, I would love to collaborate with you.
Okay,
You
yeah.
can share my email with them, with all students.
Okay, okay, sure. Sure, I will share your email with my students, and hopefully, you will get one of the potential candidates from my students. Okay, and hopefully, this webinar will open the new window for the next activities for our collaboration. And this is, I think, this is an opportunity. Okay, so we can work together in future if possible. Okay, so this is the ending of the Q&A section of our webinar, and we are very close to the end of this program. And in this state, especially, I would like to give thanks again to Agatha Maria for her time and cooperation. And I hope that all of you enjoy this webinar very much because really that was an amazing webinar, amazing presentation from Agatha
Thank you.
Maria. Okay, okay, so so and, and this webinar is basically organized by Jamal Nozrul Islam Astronomy Club, just which is a newly formed astronomy club at Joshua University of Science and Technology, Bangladesh, and we are trying to basically extend. We're trying to uh, popularize the concept of astronomy, the development of astronomy, the research of astronomy in our country. So that's why we are trying to explore the activities of this club by organizing this type of program, and hopefully we will continue this type of effort in the long run. And for this time, I just want to say goodbye and goodbye for today. So, and thanks again, Agatha, and hopefully see you in any another webinar. So, till then, wish you all the best. Okay. So, bye bye.
Thank you very much. Thank you very much. And I hope that now you can go to bed, that your melatonin levels are high enough. And one more time, thank you very much for invitation, Hassan. It was a great adventure.
Okay. So, thank you. Bye bye.
Thank you.